welcome you all. So today I have taken one question from GATE Life Sciences question paper 2014 from biochemistry section. This is question number 10 of 1 marks, 1 mark only. So here they have given that given below is the hydropathy plot of monomeric transmembrane proteins, and it has been asked how many transmembrane alpha helices are present in the protein. So by seeing this, simply you just need to know. Uh, so from here to here. So this graph represent on x-axis, it is represent first amino acid in the window. Okay. So here this is a polypeptide chain entirely that has been plotted against the y-axis, which is showing the hydropathy index. So if we'll see the y-axis, in between it is 0, and above that those are positive values, and below that the negative values are there. So here this amino acid, if we'll see, it is 140 amino acid longs. So 1 to 140, this entire amino acid structure is there, and in this amino acid they are trying to plot which region is consist of majorly of hydrophobic amino acid, which one is majorly consist of hydrophilic amino acids, like that. So that they have plotted here in the graph. So the positive value in the graph says that it has a hydropathy index in a positive number. That means that portion is hydrophobic. So the top portion is consist of hydrophobic amino acid, and the values which are in negative, those are okay, the value which are negative, which represent the hydrophilic amino acid stretch. So that is how this graph can be predicted. Now it has been asked how many transmembrane alpha helices are present in the protein. Okay, so it has been asked about transmembrane alpha helices. So transmembrane alpha helices means in a in the membrane of the protein, membrane of the cell. Okay, in the membrane of the cell. So we know our membrane is a bilayer, which is a phospholipid bilayer. This is our membrane. Okay, and then the question is transmembrane, and transmembrane means that it spans through the membrane. So the protein spans through the membrane. Okay, it spans through the membrane, so it will maybe it is we can say it is an integral membrane protein. Okay, so it can be represented this. It's an helix. Generally integral proteins are these alpha helices. Okay, okay. So as we know, in the alpha helices we have the transmembrane region of many integral proteins is made of this bundle of hydrophobic alpha helices. Okay, so uh, in the membrane bilayer, which is a phospholipid bilayer, if we'll see, so as I showed it, it is like this. is a phospholipid bilayer, something like this. Bilayer. Like this. Okay. So, if we'll see the detailed structure of this phospholipid bilayer, so this phospholipid bilayer or these phospholipids are a class of lipids whose molecule has this hydrophilic head. This is a hydrophilic head, and uh, we can say the hydro. So this hydrophobic head and two hydrophobic tails, which are fatty acid tails. Okay, one hydrophobic head and two hydrophobic tails. Okay, and these are of fatty acid, and these joined by an alcohol residues. So hydrophobic head is made of basically phosphate. So it is a phospholipid, right? So it is phospholipid. So we have the head is phosphate group. In the tail we have uh, fatty acids. So this is why it is called as phospholipids. Phospholipids. Okay, so this phosphate and fatty acid are joined by a glycerol molecule. Okay, alcohol molecule, which is generally glycerol. So if I write this will be a phosphate group. This head, which is a phosphate group, which is uh, with the help of glycerol molecule, it is joined. This is a glycerol molecule. This is a glycerol. This is the phosphate group, and this is further joined with the fatty acid tails. Okay. So this is the glycerol, and this is the tail, which is the fatty acid tail. And this phosphate group is uh, can be modified with simple organic molecules such as choline. So this can also be modified with choline. So this is the basic structure. So this portion is the hydrophilic portion, hydrophilic, and this portion is the hydrophobic portion, hydrophobic, which can be represented like this. Okay? Hydrophilic and hydrophobic. So in the lipid bilayer, when we'll see in the bilayer, the if the membrane protein is integral, it is an integral protein, it needs to be the interior portion should consist of mainly of hydrophobic amino acids so that it can stay inside this entire hydrophobic portion. This is hydrophobic and this heads are hydrophilic. Okay, hydrophilic. So this protein, so in the alpha helices, the amino acid residues which have the hydrophobic side chain in them, these side chains interact with the inner hydrophobic fatty acid chains. So this is the fatty acid or hydrophobic amino acid side chains which interacts with the hydrophobic fatty acid uh, tails inside the phosphorus bilayer. Okay, so this is was so mostly this transmembrane integral proteins are made of the alpha helices, which work as a receptor and helps in recognition of the signals. So here if we'll see how many question has been asked according to this graph, which is the hydropathy index, how many transmembrane alpha helices are present in the protein. So if we'll see the this side which shows the hydrophobic amino acid stretch, so here we can see two peaks. One peak is this one, another peak of this one. So here as we can see two peaks, we can say that there are two integral amino acid. So this is one pass, a single pass transmembrane protein I showed. It can be like this, that instead of going like this protein again spans. This is could be a multiple pass protein as well. So either it is a single pass, or if more alpha helices or more hydrophobic alpha helices are there, it could be a multiple pass integral transmembrane integral proteins. Okay, so here we can see two transmembrane alpha helices could be present, but if we'll see the distance properly, so here from 20 to 40 amino acids are showing a hydrophobic stretch. So 20 amino acids. Here it is very less. So the protein to make an integral alpha helix to be part of the membrane, it needs to have at least 20 or more than 20 amino acid. So here this particular stretch does not qualify this criteria. So based on that, only this peak which shows the 20 amino acid hydrophobic stretch in this particular protein shows that there could be only one alpha helix which could work as a transmembrane alpha helix. So out of these four options, the option A, which shows only one transmembrane alpha helix, is the correct option. So this won't be considered as a transmembrane alpha helix because very short stretch of amino acid residues. This is my NPTEL reference.